Hello everyone, and welcome to the first lecture from chapter 17, which is about the citric acid cycle. We're going to start this first lecture actually right before the citric acid cycle, where we're going to discuss the pyruvate dehydrogenase complex. Overall in this chapter, we're going to talk about the pyruvate dehydrogenase complex, the citric acid cycle, and how both of them are regulated. So first, let's sort of orient ourselves in terms of where we are in glucose metabolism. So we discussed in the previous chapter glycolysis, how glucose is converted in one molecule of glucose is made into 2 molecules of pyruvate. The next step in aerobic respiration after pyruvate is produced is the citric acid cycle. But to get there, first we have to, first let's talk about what exactly the citric acid cycle is. So in the process of the citric acid cycle, there are 6 carbon dioxide molecules produced for every glucose molecule. So each of those 6 carbons gets fully oxidized. And energy is going to leave the citric acid cycle as the reduced electron carriers, NADH, FADH2, as well as GTP and ATP. The citric acid cycle is also really important for amino acid metabolism and fatty acid metabolism. And as you can see in this diagram, there are, from protein metabolism, as you break them down, there are different entry points that can ultimately lead to the citric acid cycle, as well as from the breakdown of various fatty acids. It's also really important or interesting to note that there are various intermediates that are produced during the citric acid cycle that can actually be sources of biosynthetic building blocks that are used to build up different important biological molecules during anabolism. So that being said, to enter the citric acid cycle before pyruvate can start going through the citric acid cycle, pyruvate has to be converted to acetyl coenzyme A. During that process, 1 carbon is lost as a carbon dioxide molecule, and then 2 more carbon molecules from each pyruvate are able to then go on to enter the citric acid cycle via acetyl-CoA. And we'll talk about this when we talk about the pyruvate dehydrogenase complex. It's also important to note that all of this is happening in the mitochondria. So all the pyruvate that's generated by glycolysis in the cytosol has to be transported into the mitochondria before any of this can happen, because all the enzymes that are helping carry out these reactions are located in the mitochondria. So with that, let's start talking about the pyruvate dehydrogenase complex, which we can also call by its initials, the PD. So the pyruvate dehydrogenase complex, like I just mentioned, is the link between glycolysis and the citric acid cycle, where we can see in this overall balanced reaction, we have our pyruvate, our last product of glycolysis, which has 3 carbons, is being converted into an acetyl-CoA, where the acetyl group now has 2 carbons. So during the process, pyruvate, which has 3 carbons, is losing 1 carbon as a carbon dioxide molecule. The other 2 carbons go on to attach themselves to a coenzyme A to become acetyl-CoA. During this process, we also have the reduction of 1 NAD plus molecule into NADH. And NADH, as the reduced form, can also be used later on during oxidative phosphorylation as another way to generate ATP. So there are actually 5 different reactions that go into this overall balanced reaction. But all of them take place on the pyruvate dehydrogenase complex. And it's called a complex because it's large and it has multiple enzymes within it. And there are 3 different main subunits called E1, E2, and E3. And each of those different subunits has a really important part to play in making sure that this overall balanced reaction can happen, as well as making sure that all of the different components of the enzyme and the active sites are reset back to their initial state once the reaction is catalyzed. So let's start talking about the different reactions that we're going to see in the pyruvate dehydrogenase complex. Before we start discussing that, I do want to mention that you are responsible on the exam for knowing the overall balanced reaction and the overall function of the pyruvate dehydrogenase complex. You will not need to know the specific individual reactions. That being said, I think it's really helpful to see how they all work. And so that's why they're in this video. And I hope you get something out of it, and I hope it's helpful for you to understand the importance of the PDC and the reaction, the overall reaction that it catalyzes. And it's also interesting, I think, to see the different sort of vitamins and minerals that we know as part of our diet and how they are functioning here in the PDC. So that being said, reaction 1 is starting in the E1 subunit, where the E1 subunit contains thiamine pyrophosphate, or TPP, and this is a prosthetic group, meaning it's permanently attached to the enzyme, to the E1 subunit. This TPP forms a covalent complex with pyruvate. So you can see in this reaction, this carb anion in TPP attacks this carbon in pyruvate. There is the loss of a carbon dioxide from the left, and the carbons on the right are then now covalently attached to make this intermediate hydroxyethyl TPP, which is permanently attached to the E1 subunit, TPP, as I should say. And now we've had this carb anion here on the molecule that used to be part of the pyruvate. The next step now is involving now the subunit E2. So E2 has a lipoamide arm, which is a prosthetic group that's part of E2. But as you can see, it's this really long hydrocarbon chain. On the end of the hydrocarbon chain is this disulfide bond that's going to help participate in the reaction. And this arm in general is very long. So even though it's on the E2 subunit, it actually reaches over towards the active site of the E1 subunit where our TPP complex is. And at this point, the carb anion from the molecule that used to be our pyruvate can now attack one of the sulfurs in that disulfide bond, and helping to partially reduce that lipoamide arm. In the process, the TPP is released. So now the E1 subunit is basically reset, where the TPP is prosthetic. This prosthetic group is permanently attached to the E1. And now our red little molecule with our 2 carbons is now an acetyl group that's attached to this lipoamide arm that's part of the E2 subunit. So at this point, we are ready for reaction 3. So now the lipoamide arm returns to the active site of the E1 subunit. In the active site, there is a coenzyme A. The coenzyme A is in the reduced form, which is indicated here by the SH. So this is just a regular coenzyme A, and it's written as CoA-SH as an indication to you to understand that it's reduced. And it's able to attack the acetyl group to form a nice little bond here where we've formed an acetyl-CoA. And the lipoamide from the E2 has been released. But now the lipoamide arm has these 2 reduced sulfurs instead of what it was originally, which was that oxidized disulfide bond. So now we've created acetyl-CoA, which is one of the products of our reaction, but in the process now we have our reduced dihydrolipoamide, which has to be reoxidized back to its original state so that the PDC complex can be reset to its original state. So so then that brings us to reaction 4. This is going to happen in E3. So in the E3 subunit, there is another disulfide bond, which is in the oxidized state. And it is able to then exchange electrons with those reduced thiol groups in the lipoamide arm. So ultimately now the lipoamide arm has been oxidized back to its original state, but now these 2 thiol groups in the E3 subunit are reduced. So those now themselves have to be reset back to their original oxidized form. And that's where the other component in the E3 comes in, which is the FAD, which is a prosthetic group that's permanently attached within that E3 active site. And you may be familiar with FADH2 as an electron, or FAD, I should say, as an electron carrier in the oxidized form, where it's able to accept electrons. And that is what happens here. So from these 2 thiol groups, those electrons are donated to the FAD, ultimately converting those 2 thiol groups back to their original oxidized disulfide form and reducing now the FAD to the FADH2. So at this point, now all that's left to do is to return the FADH2 to its original oxidized state, and that happens with the help of another electron carrier that you may be familiar with, which is NAD+. So at this point, NAD+ accepts those 2 electrons from FADH2, generating an NADH and a proton. Now that NADH is now able later on to be used in oxidative phosphorylation to help generate ATP. And now the FAD is reset to its original state, and so now the E3 subunit is finally reset to its original state, meaning that finally the PDC is done catalyzing that very complicated 5-step reaction. So this figure helps to show sort of overall what just happened. We had, from one glucose molecule, we formed 2 pyruvates. Those pyruvates, each pyruvate molecule loses 1 carbon in the form of carbon dioxide. The other 2 carbons attach to coenzyme A to create acetyl coenzyme A with that really high energy thioester bond that we'll see come into play later on. And also we reduced this NAD+ to an NADH, which can also be used later on during oxidative phosphorylation. So overall, if we had 1 glucose molecule, we essentially ended up now with 2 acetyl-CoAs and 2 NADHs. So as a summary in words, this basically says what was shown in pictures on that previous slide. For each 1 pyruvate molecule, 1 pyruvate is fully 1 carbon is fully oxidized to CO2, 1 NADH is produced, 1 pyruvate molecule is converted into 1 acetyl group, and it goes from 3 to 2 carbons because we lost that 3rd carbon as a CO2. And finally, our acetyl-CoA, which contains that nice high-energy thioester bond, is ready to enter the citric acid cycle. And remember that all of this is happening twice per glucose molecule. So with that, now you should have an understanding of the pyruvate dehydrogenase complex, and you are ready to answer the practice questions.